what we're going to do for most of the class today is continue on the tour of microbial diversity, and that will give us the background information that we will then use for next week's lectures, where we're going to talk about functional diversity, phenotypic diversity, and as I mentioned, sort of other aspects of how microbes interact with the world and with each other and with people. We're going to basically go through a few different examples in the next few lectures about the lives of microbes. So what we're going to do today, again, is continue on this tour. I know I started in on the eukaryotes. I'm just going to back up a little bit for the first group that I already covered. Go through really 5 different groups of eukaryotes in detail, and then the last 2 are primarily going to be covered in later classes. And then we'll talk about a little bit more detail on endosymbioses, which we've already talked about a little bit, and then if we get to it at the end, lateral gene transfer. And both of those are examples of, in essence, where we don't have a single phylogeny for individual organisms. There are multiple crossing branches in the tree. All right, so this is the general, very coarse, oversimplified phylogenetic tree of eukaryotes with these 7 major lineages: Alveolates, Stramenopiles, Rhizaria, Excavates, plants or Plantae, which mostly we'll talk about later in the class, Amoebozoans, and then Opisthocons is this last group, and mostly we'll talk about that later because that includes the fungi and the animals. So I've mentioned this already, but I'm just going to talk about it again. When we look at the evolutionary relationships among bacteria and archaea, most of the information that we have about the evolutionary relationships there is based upon molecular data and usually DNA sequence information of a variety of genes or genomes. And in eukaryotes, that's also true, but as you will see when we go through the different major eukaryotic groups, there's a lot more morphological diversity in many of these groups, and that morphological information as well as physiological information has been very useful in the phylogeny, in inferring the relationships among these eukaryotes, although we will go through some examples of exceptions. One reason that's really important is not just for studying modern organisms, but it turns out that the fossil record for microbial eukaryotes is very useful, in part because the phenotypic morphological information is informative. So if you find a fossilized eukaryote, you can, single-celled even, you can frequently say what type of eukaryote it was, whereas with bacteria and archaea, that's much harder because, again, phenotype is not that useful. So get ready. Going to continue. We already sort of talked about this. I'm just going to go through it again and have a clicker question related to one of these. So the alveolates are one of these 7 major groups of eukaryotes. Most of these 7 groups were identified based upon molecular phylogenetic analysis or some other type of phylogeny of a large number of traits. Many of them people have identified synapomorphies that are shared by members of those groups and appear to be shared derived traits for those groups. So the alveolates, they have these sacs, also known as alveoli, that are beneath the surface of the plasma membrane. And that's a defining feature of the entire group here. All of the members that have been studied in these groups, in the alveolates, of which there are 3 major lineages, are single-cellular throughout their life cycles. And many of them, although not all of them, are photosynthetic. So So just going to go through, again, I briefly touched on this. These are the Dinoflagellates, one of the three major lineages of Alveolates. Most of them are marine. They are very important to primary production on the planet, that is the fixation of carbon. They contribute a significant amount to the total fixation of carbon on the planet. They have these diverse pigments in them, of which many of them have this sort of brownish tinted color. And again, the defining feature of these group are these two flagella that wrap around the outside of the cell, one of them in this equatorial groove and one of them in this longitudinal groove. Just as background, little videos here. They have really cool behaviors in when you observe them in the wild or in the laboratory, and there's a lot known about the biology of some of these species because they can be reared in the lab. Some of them have not been cultured, that is, grown in the lab, and we know less about them. So here's a clicker question to welcome your Friday. So the Dinoflagellates, many of them are photoautotrophic. What is the difference between photoautotrophy and chemoautotrophy? And I'll give you till 1:30. All right, 5 seconds. All right. Phew, either you looked it all up in your notes here or you remembered, but you should know the different types of trophies here, the source of energy, the source of carbon, the source of electrons, and in this case, photo versus chemo, that's the first part of the panel I showed before. That's the source of energy that organisms use, and for these dinoflagellates, many of them use the energy of light and are therefore phototrophic in some way or another. All right, so just going to, again, continue on through the alveolates. The apicomplexans, all of the species in this group that are known are parasitic. That is, they negatively affect some other organism. We'll talk about the definition of parasitic in more detail in one of the next two lectures. The name of this group, the apicomplexans, comes from this mass of organelles that all of the species in this group have at the tip of one of the cells. It's called an apical complex, and that's where the apicomplexan name comes from. This is a shared trait of the members of the group. The best known member of this group is our Plasmodium species, in particular Plasmodium falciparum, which is the causative agent of many of the forms of human malaria. There are other species of Plasmodium that also cause malaria, and then there are many relatives of Plasmodium that also cause disease in various plants and animals. The book has background information on the Plasmodium life cycle. It's really kind of cool how it goes through multiple stages when it infects, gets into mosquitoes, and then is transmitted into humans or animals, and then undergoes various sexual or reproductive strategies. And then the third group of the alveolates are the ciliates. I've had in the slides that I think I posted, it says the structure is identical to flagella. I've crossed that out here. It's not identical. It's very similar in the core parts of the structure, cilia and flagella, but they are not identical structures. So this group is defined, all of the members of this group have these small hair-like projections on the outside of the cell that they use for both movement and for feeding. So they can pump fluid into compartments that are sort of exposed to the outside of the cell, and then they can envelop those compartments and feed off of them. So they use the cilia both to move the cell and for this feeding. And all of the members of this group have a really strange component of their cell biology, which is that they actually have two distinct nuclei. Rather than most eukaryotes have a single nucleus where they keep the genomic DNA of that organism, the ciliates have this really strange thing where basically one of the nuclei is the equivalent of what we see in animals of the germline. That nucleus participates in sexual reproduction and not much else. And the other nucleus is sort of the equivalent to the rest of our body, the somatic cells. And that nucleus is what's used for normal sort of expression of genes and other life cycle stages. And there's incredible morphological diversity among the ciliates. There are thousands of species that have been characterized largely based upon their morphology. So now we're going to switch to group number 2. The book in chapter 27.2 and a little bit in some of the other sections of 27 basically goes through very similar material to what I'm covering here. Not exactly the same, but many of the examples I'm trying to use are similar or the same as are used in the book. So if you want to get extra information or remind yourself of what we're talking about today, the book covers basically these same lineages. So this lineage is called the Stramenopiles. They have a synapomorphy that they share with each other, which are these two flagella, but then these rows of these tubular hairs on the longer of the two flagella. They're really kind of cool if you look at them, the cells of these organisms in a microscope. And then there are 3 main lineages that we're going to talk about: the brown algae, the diatoms, and the oomycetes. The brown algae, you may not know about the group, but I'm sure you know about some of the organisms in the group. Giant kelp is a member of this group. So many of the organisms in this group look a lot like plants. All of them are multicellular, and they phenotypically resemble, in many ways, various types of plant species. Those similarities are due to convergent evolution. The ancestor of the brown algae lineage was a single-celled organism. So all of these similarities with plants are due to convergent evolution. Some, like giant kelp, get very large. Some are quite small. They're almost exclusively marine, which means they're in salt water, not fresh water. And the brown color of a lot of them comes from this particular type of pigment. We'll talk more about them when we talk about the evolutionary history of chloroplasts, because they have a really strange history behind they are photoautotrophic, they have chloroplasts, but they have this unusual history of their chloroplasts. The second major group of Stramenopiles are the diatoms. You may have seen on and off some pictures of them. They're really fascinating structures on the outside of the cells. They have an incredibly good fossil record, so we know an enormous amount about their past history. Their fossilization is also a major source of oil and gas, so the degradation of diatoms after they die over millions of years has led to the buildup of oil and gas in various fossil beds. They're generally single-celled, but many of them, like the one shown in this video that I'll show again here, form filaments of many cells stuck together. I think most, if not all, that have been basically studied are photoautotrophic. That is, And the estimates are that they actually, globally across the planet, contribute up to 20% of all the carbon fixation that happens on the entire planet. So very important from an ecological point of view and from a variety of other points of view. And then the third group of Stramenopiles are very strange, starting to be better understood, but still not as well understood maybe as diatoms and brown algae. They're no photosynthetic, photoautotrophic members of the group. All of them are what are known as absorptive heterotrophs. We will talk about that property again when we talk about fungi, because fungi are also absorptive heterotrophs, and they have many phenotypic similarities to fungi, but they are not closely related to them. They are in this Stramenopiles group, very distantly related from the fungi that we will talk about later in the class. The causative agent of potato leaf blight, which was responsible for the Irish potato famines and now this sudden oak death that's going around California and other locations, is a member of this group. You can't really see this that well, but that's a Phytophthora species, which is one of the species that causes these diseases. All right, another even more exciting clicker question. So the similarity in appearance of oomycetes to fungi is an example of what? And I'll give you till 1:15 this time. All right, 15 seconds or so. Last chance. All right, good again. That is the correct answer, C. So this is an example of homoplazy because the origin of these phenotypic similarities, the similarity in appearance, arose separately in evolution, two different lineages, and therefore it's an example of not of homology, not of common ancestry, but this term that I talked about called homoplazy. Are there any questions about this? Most people have got it right. So, okay. All right, so continuing on the tour here. The third group we're going to talk about is known as the Rhizaria. They're all single cellular. I think all that are characterized are aquatic in some way. I'm not 100% sure of that. And they have these long, thin pseudopods, which you'll see some pictures of, a few of them in a second. And there are three main lineages. For most of these groups, there are many, many more lineages than we are talking about here. We are talking about here the ones that are either best understood or that have major impacts on the planet or humans in some way. But there are other lineages that are nestled within these that we're just not going to talk about. So again, these are the three major lineages. The Cercozoans are really quite poorly understood. There are very few of them that have been characterized in detail. This is a picture, an electron microscopy picture of one of them. They've been found in ponds, in soil environments. They're found in lots of different places, but they're generally not very abundant, or they can be hard to rear in the laboratory, so we don't know a lot about them. One group in this lineage, and we'll talk about this again when we talk more about chloroplast evolution, is photoautotrophic, and they have chloroplasts that they have inside of them. The second major lineage within this group are known as Foraminiferans. You may not be familiar with the individual organisms, but you probably almost certainly are familiar with many of their outputs. So in many places, the sand on beaches, like this pink sand beach, is the result of sort of dead foraminiferans. Their discarded shells also make up limestone. And there's a lot of information about their fossil record, again, like many microbial eukaryotes. And that fossil record has been used in many cases to provide not only information about their evolutionary history, but even about the age or the particular sort of history of different bands of sedimentary rock as you identify different foraminiferans in them. Again, they have these pseudopods, thread-like appendages that you can sort of see in this picture, and they use them to feed. They're coated in a mucousy-like surface, and it makes this sort of really sticky net that they collect other organisms from their environment and then eat them. And I just thought I'd show this here. This is a foraminiferan. It's about the size of your fist, and it's a single cell. It has many nuclei that are present, but there's no division between the different cells. It's a gigantic single cell found in the bottom of the ocean. There are many of these gigantic foraminiferans that are found on sediments in the bottom of the ocean, and you can actually, when they've sent down submarines down to the bottom of the ocean, you can see tracks. They actually sort of crawl along the ground, and you can see tracks behind this sort of giant single-celled organism. And I'm going to show, I hope here, this is, here I'll just tell you before, this is a video from the Alvin submarine, a deep-sea submarine from a research trip that I went on. I wasn't in the submarine, but I was on the surface of the boat. And there was a graduate student from Woods Hole Oceanographic Institute who got to go in this dive in the Alvin submarine 2 miles down in the bottom of the ocean. Normally, it's hard for graduate students to get into the Alvin because everyone else wants to go, and all the faculty and other dignitaries are fighting all the time. And so she got to go down, and she had been spending a year or two curating a collection of information about all the diverse organisms that had been observed in Alvin dives previously. And they're cruising along in the submarine. I don't know if people know the Alvin, but it's what found the Titanic, and it's a deep-sea submarine that's been used for a lot of research for about 50 years now. And it has a big submarine sort of body, but then there's this little 6-foot diameter titanium sphere that the 2 or 3 people sit in, and there's a pilot and then usually 2 scientists. And these tiny little windows that are like 6 inches thick glass to make sure they don't crack, that you're peering out of as you cruise around in the bottom of the ocean. And they were doing this, and she's looking out the window basically, and they're seeing weird, fish and other things, and then this shows up in the window. And the other people in this cruise had no idea what this was. And she immediately knew that it was, because she had been curating this information, that it was this xenophyophore, really weird, giant, single-celled organism. And she collected it, and I have it in the freezer. We've been trying to do some genomic analysis of this organism because it's really, really bizarre and strange. Anyway, nobody apparently wanted it after she collected it, and I was the dorky microbiologist there saying, I will take it home with me. And if you Google xenophyophore, I was not responsible for this, but if you Google xenophyophore, this is actually the picture in Wikipedia is of the actual organism that we have in the freezer. All right, then the third major lineage of the Rhizaria are the Radiolarians. Also really fascinating cell structures. They secrete these glassy skeletons with incredibly elaborate designs. I don't know if this, and they also use these pseudopods. In this case, I think they mostly use them to sort of create large surface area to acquire nutrients from the water, and they use them a little bit for as a flotation device, basically. They're also, like many of the other interesting morphology, microbial eukaryotes have a pretty good fossil record. All right. Next group, now with, for better or worse, 5 major lineages, although really 2 of them we're going to talk about together because they're generally considered. These 2 are distinct lineages, but most of the time they're sort of lumped together because they have one really interesting property that they share in common. And that is, so this is the Diplomonads and Parabisalids. An example that you may have heard of is Giardia. Giardia is one of the Diplomonads. Causes really unpleasant stomach, gastrointestinal problems that you don't want to get. Places in the East Coast in particular, it can be transmitted into water by organisms like beavers and other species, and it's one of the reasons why in many places you want to filter, sterilize, or otherwise sterilize your water because you don't want to get this. Trichomonas vaginalis is a parabisalid. It's a sexually transmitted disease. And what's most interesting about these, this is a little video of Giardia swimming around. What's most interesting about these lineages is that they do not have mitochondria. So as we will talk about when we talk about a little more about symbioses, analyses that people have done have indicated that the origin of mitochondria happened at the beginning of eukaryotic evolution, and therefore eukaryotes, they have a nucleus. Eukaryotes that do not have mitochondria have lost those mitochondria after early events in eukaryotic evolution. So there's some really interesting studies of these taxa to try and understand what happened after they lost their mitochondria and what they, in essence, do to compensate for the lack of them. All right, so the next group of the excavates are the Heteroloboseans. You, again, may not be familiar with the group, but you are almost certainly familiar in the press with some of the organisms in the group. So one species of this Naglaria is the brain-eating amoeba that gets press every once in a while. It can be found in municipal water supplies or in lakes and ponds, and occasionally, I mean, it doesn't kill that many people in the country, but it's very unpleasant when it does, and it usually gets some, horribly sensationalistic press associated with it. Many of them can transform between these amoeboid, which are just basically blob shapes, and these flagellated shapes with, flagella that they swim around with. And like with the chlamydia example that I gave you about bacteria, many of these have been misclassified as 2 distinct species when people observed the amoeboid form versus the flagellated form. The second to last group of the excavates are the euglenids. We will talk about them again in a little more detail when we talk about the evolution of chloroplasts and photosynthesis. Some of them are photosynthetic. Some of them are heterotrophic, and some of them can switch back and forth. That's relatively unusual. Many photoautotrophic species aren't very good at acquiring energy outside of their photosynthesis much of the time, but many of these species are pretty good at doing both forms. And then the last group of the excavates are the kinetoplastids. Again, you probably are not particularly familiar with the lineage here. The lineage is defined by the presence of this particular kinetoplast, which is a structure within the mitochondria of these species, and sort of defined by microscopy. But the causative agent of many diseases of humans and animals are within this group. So Chagas disease, sleeping sickness, leishmaniasis cause lots of problems, in particular in tropical regions of the world. They're transmitted generally by some type of insect. The particular insect that transmits these varies between the different parasites, but there's a lot known about this group because they cause diseases in humans and animals that we care about. All right. Moving on to One of the last lineages we're going to talk about, the Amoebozoans. These are, as the name suggests, amoeboid much of the time in their cell form, that is blob-like. And there are three major lineages. There are two that generally are referred to as slime molds, but they now have two different major subgroups within these that are evolutionarily distinct from each other, although they are sister taxa. And I'll show you a little bit about them. Loboseans, this is a photosynthetic Lobosean crawling around here. They almost I think actually all of them that are known live as single cells. There are many pathogens in the group. They secrete these sort of glue-like structure on from the outside of the cell, and they can create like a house around them, kind of like you might have seen with various, hermit crabs and other animals. These single-celled organisms can sort of collect various materials on the outside of their cell with this glue-like substance. And Loboseans were in the press yesterday, actually. There's a really cool story, or scary story if you want, about this particular Lobosean, which actually causes dysentery and is a major human and animal pathogen, Entamoeba histolytica. It's, they did really fine-scale microscopy of this organism, and it's this one in green eating red blood cells, and I have some videos here where they reconstructed. What it does is it basically goes in and bites off part of the host cell, and this has basically never been observed in any member of this group before. They did this three-dimensional reconstruction. It's basically chewing on gut cells. That's how it's causing dysentery. It basically pops them open by biting off a part of the cell. And there were I heard it on the way home on NPR, so I figured should show it to you. So this is one of the slime molds. These, after rainfalls, you frequently can see some of these around Davis growing out on bark chips and other places. This is a very sped up, it doesn't move this fast, probably 100-fold speed up, but they do literally crawl around, gliding on the slime that they secrete. Makes it much scarier when you think they move fast, but... and what's really interesting about many members of this group is that they can get together, individual cells can get together, and that's what this sort of crawling across the surface is. It's a collection, a sort of colony of cells, or occasionally it's also a single cell, like that giant xenophyrophore that I was talking about, that has many nuclei, but just one cell membrane around the cells. And again, many of them create these fruiting body structures that are multicellular. And then the cellular slime molds have many similarities. Again, they're a distinct evolutionary group. They are, the one member of this group in particular, Dictyostelium, has become a model organism for experimental studies of eukaryotic cells as well as social behavior in eukaryotes. And this is a video of Dictyostelium as the individual cells start to collect together to eventually generate these aggregated amoebas, and then it's going to form a fruiting body in a little bit here. They're really become a really interesting model system for understanding why cells might work together to form. I'm just going to zoom along here. So now they've blobbed together and they're going to start to grow these fruiting bodies, and then that's their reproductive strategy. Those fruiting bodies eventually release spores that lead to new generations. All right, so that's the 5 groups that don't include plants or fungi and animals. And I'm just going to quickly give you an introduction to the plant groups and to the group, the Epistheconts, that fungi and animals are in. We're going to talk about these in more detail later in the class. So if we look at the plant lineage, there are 5 major lineages: land plants, and you'll hear more about charophytes when we talk about land plants, and then 3 lineages of basically other types of photosynthetic eukaryotes that share a common ancestry with land plants. The first lineage of which there isn't a lot known about many members of this group, although a few are well studied, are called the Glaucophytes. And the chloroplast in the Glaucophytes has peptidoglycan in it between an inner and outer membrane. That should tell you something about the history of these chloroplasts, and that will be the basis of another clicker question related to peptidoglycan. So, which of the following groups do not have peptidoglycan in their cell envelopes? Till 1:15 here. All right. Ready? Everybody got answers in? How about now? All right. I'm going to stop. All right. Good. C is the correct answer. Although not quite as many as got it correct as the other answers. Are any questions about this? Peptidoglycan is a feature found in all bacteria except this one lineage that I mentioned, the mycoplasmas that have thrown away their cell wall, but all other bacteria have peptidoglycan as part of their cell wall. It's a distinguishing feature of bacteria. It is not found in archaea. And the only time it's seen in eukaryotes is when it's seen in an organelle, that is, an evolutionary history of coming from bacteria. So finding peptidoglycan in Glaucophyte chloroplasts is one of the lines of evidence that supported the theory that chloroplasts are evolutionarily derived from bacteria because no other lineages have peptidoglycan. So it's sort of a hallmark feature that indicates where the chloroplasts came from in this case. Quickly, I'm just going to introduce the other plant lineages. The red algae, you've probably seen many of these. Many of them are marine. They can wash up on the shore in many marine environments. Many of them are multicellular, although not all of them. They are photoautotrophic. They have this red pigment rather than a green pigment that defines their sort of photosynthetic apparatus, and that's why they're known as red algae. And then the chlorophytes frequently are referred to, or by some, as green algae. They have many of the components that they have in their photosynthetic apparatus are similar to those seen in the charophytes and land plants. Incredibly diverse group. There are at least probably more than 20,000 species that are now described, and you're also probably familiar with them. You see many multicellular versions of this, again, in marine and sometimes freshwater environments. We'll come back to land plants and charophytes when we talk about plants in a few weeks. And then the last group, I'm just going to talk about the Dinoflagellates quickly. The last group is known as the Opisthecons, and it includes the fungi, the Dinoflagellates, and animals. This grouping of these three taxa is important for many reasons. So it means that studies of fungi can be used, they're not very closely related to animals, but fungi have more synapomorphies, more traits that they share in common with animals than animals do with many other single-celled eukaryotes. So it makes studies of fungi a little bit more relevant for understanding animal biology than, say, studies of Plasmodium. And then, interestingly, there's a lineage of single-celled eukaryotes, now known as Dinoflagellates, that is actually more closely related to animals than fungi are. So this is known as, generally as the sister lineage to animals. They have these cells that we'll talk about when we talk about animals that resemble cells found in sponges, and that's where they get this, the sponges get the name for their cells. You'll see that later when we talk about animal evolution. Many of them are colonial, some of them hang out as sort of single-celled organisms. But they've become a really important model system for studying animal biology in a very simple system because they're the sister group to animals. And again, fungi and animals we will cover later in the course. So what I want to do in the last few minutes here is start to re-talk about one of these major complications in the evolutionary history of eukaryotes. Well, actually, the evolutionary history of everything, because they're bacteria that are moving inside eukaryotes via these endosymbioses. And again, if you look at this on the tree in the course, from the book, they tried to draw that there was this symbiosis with proteobacteria that led to mitochondria. After that, there was a symbiosis with cyanobacteria that led to the chloroplasts. This mitochondrial event happened prior to the existence of the common ancestor of all modern eukaryotes. This chloroplast event, as we will see later, happened prior to the existence of the common ancestor of just the Plantae lineage. And again, after the mitochondrial event, that's when the diversification of these 7 major lineages of eukaryotes occurred. And there are many organelles that are seen inside eukaryotes. An organelle is basically a membrane-bound compartment. We're focusing largely on sort of the evolutionary history of these 3 different organelles. The nucleus is where the genomic DNA of whatever the host cell was prior to these symbioses. The mitochondrion is, again, this proteobacteria. The chloroplast is derived from cyanobacteria. There are many other organelles inside eukaryotic cells. Most of them do not appear to have a symbiotic origin. They appear to have originated by changes that occurred inside the cell of eukaryotes rather than via some type of symbiosis with some other lineage. And I'll just go through this sort of animation that came from the people who made the book, and I'll try to post this online. I'm not sure how well that'll work. But basically, this is a model about the events in the history of eukaryotes leading up to the modern organelles in eukaryotes. So the common ancestor was some type of prokaryotic cell. As I mentioned before, it may have been from this one lineage of archaea, or it may have been a totally separate group that led to the common ancestor of eukaryotes. This lineage had some type of cell wall. We would infer that because archaea and bacteria both have cell walls. They're different, but the most parsimonious explanation is that the common ancestor of everything had some type of cell wall. So early in the origin of this lineage, it had a cell wall, had DNA packaged somewhere inside the cell, but then it lost the cell wall. That's what led to the lineage that eventually became the ancestor of the nucleus of eukaryotes. And the reason that people think it lost the cell wall is it has to carry out this bringing in the bacterial cell inside of it, which only works really well if you don't have a cell wall. So after it lost the cell wall, there were all sorts of other things that happened to make the cell a little more complex. This may have been what led to the creation of things like the endoplasmic reticulum and the Golgi apparatus and other hallmark features of the eukaryotic cell. I couldn't delete this animation. So then after this, there were more events in the history of the eukaryotic cell. At some point, a cytoskeleton evolved. These are the holds together eukaryotic cells and is an important part of the biology of eukaryotes. Cytoskeletons are used both to provide structure and to move around different parts of the eukaryotic cell. Again, more internal membrane formation. Continued evolution of complexity of this cell until it gets to some point where it's able to have this ability to carry out phagocytosis. That is, like an amoeba-like cell that I showed you some pictures of. It's going to be able to feed on other components by wrapping the membrane around it. And at some point, it evolves some interaction with, this should really say proteobacteria, with a proteobacterial lineage that was eventually brought inside the cell. And that's what became the mitochondrion. And this, a little bit after this event, you have now a mitochondria that evolves. It throws away its cell wall. And this, before it evolved, before any lineage evolved in interaction with cyanobacteria, this cell with a nucleus and a mitochondrion and some other complex features like a cytoskeleton and these internal membrane structures, that is the common ancestor of all eukaryotes. The 7 major lineages of eukaryotes diversified from this, a cell like this. As part of that diversification, one of the lineages of eukaryotes, the common ancestor of the Plantae lineage, evolved an interaction with a cyanobacteria, not yet a chloroplast, a free-living cyanobacteria. That was also brought inside the cell. This is now just in the plant lineage, and that led to the evolution of chloroplasts. And so these 2 major symbiotic events, mitochondria in the common ancestor of all eukaryotes, and then chloroplast in the common ancestor of all Plantae, are critical events in the evolutionary history of eukaryotes. And we will continue to talk about this in some subsequent lectures. Hold on, I'm not done yet. Another really important thing that wasn't really highlighted in that series of animation that I want to highlight here is that the timing of these events is important to think about what was going on in the other lineages of life. So if you imagine the common ancestor of everything on the planet, and if we believe that rooted tree that I showed you, there was a split into 2 lineages. One of them eventually led to the common ancestor of archaea and eukaryotes. One of them led to the common ancestor of bacteria. In blue, I'm drawing bacteria here. The bacteria started to diversify into those many different groups of bacteria, including the ones we talked about, the 6 major lineages we talked about. The lineage leading up to the common ancestor of archaea and eukaryotes still may have diversified, but we don't see any of those lineages left today. Eventually, the eukaryotic lineage and the archaeal lineage split, and now you have the common ancestor, a common ancestor of archaea and the eukaryotic nucleus at least. Bacteria continue to diversify. Eukaryotes have not, we don't see any descendants yet of other lineages of eukaryotes, so this is a common ancestor of all eukaryotes. And it's this that engaged in a symbiosis with the proteobacteria that led to the mitochondrion. So when this symbiosis happened, and then eukaryotes diversified, there were already many, possibly hundreds, of diverse lineages of bacteria, and many, although maybe not hundreds, of lineages of archaea that were already present. And that's what allows us to then pinpoint that the mitochondrion came from within one particular lineage of bacteria, the proteobacteria. Those lineages were already diverse on the planet, and one of them led to the common ancestor of proteobacteria. And then I'm not showing it on here, another lineage, the cyanobacterial lineage, led to the chloroplast. And we will talk about that evolutionary history in the next class.